Hi there, everyone. So welcome to March's UK International Coronavirus Network's online seminar series. So we've got two fantastic speakers today, Professor Gary Whittaker from Cornell and Dr. Helena Meyer from the Pirbright Institute in the UK. Just a reminder to everyone that the talks are recorded and they'll go up to YouTube after. It's just the talks rather than the questions that are recorded. It's approximately 20 minutes for each talk, and there'll be some opportunity to ask questions. This is opened up as just a normal sort of Zoom meeting. So if you'd like to ask a question after the talk, just raise your hand and then I'll come to you in turn. So without further ado, if I can hand the floor to you, Gary. So I just lost my screen for a second there. So Professor Gary Whittaker from Cornell is going to talk to us about coronavirus spike protein-mediated membrane fusion and cell tropism. Okay, Gary, over to you.
Okay, are you seeing my title screen?
Yes, we can do.
Okay, right. Thanks, Dalan, and thanks for the opportunity today. So what I decided to do today was, I know I was in this group back in May last year in Belfast. So I'm going to do, just so you know, a little update on what I talked about, streamline things a little bit, an update. And with the title on the screen here is we've got a big focus on feline coronavirus right now, but with the subtitle, I'm starting to think, as you'll hear toward the end of the presentation, about the situation in Cyprus, thinking a little more widely about this group of viruses, how they link to SARS-CoV-2, and how, what's really going on in terms of this apparently very dynamic world of what I'm calling a mix of mutations, indels, and recombination to drive this transmission and disease process and this bunch of really interesting coronaviruses or companion animals. So I'll jump right in and start with start with SARS-CoV-2 because everything I'm going to present sort of is really grounded here. It's grounded to some extent in influenza HA, and I'll mention influenza in passing along the way, but really for the sake of today, we're focused more on SARS-CoV-2. And as many of you know, I'm not sure I have a pointer, actually. You're not seeing my pointer. I'll try and describe and not point. Sorry about that. So SARS-CoV-2 then has this insert, right, which I'm sure most of you are aware of. So SARS-CoV-1 has a cleavage site, but it's not this so-called furin cleavage site, which is what got inserted into this at some point into SARS-CoV-2 as it emerged out of its reservoir. So we have an unusual 4 amino acid insert, proline, arginine, alanine, sitting in this position to create this new cleavage site, which became a really big factor in the the origin of the virus, the transmission of the virus. And I'm summarizing a lot of work here in one slide, but the way I see the whole relevance of the furin cleavage site is it's really allowing these up conformations of spike. We now know, based on a lot of very nice structural studies from a lot of people, we have a very dynamic spike protein. It's very unusual for a viral glycoprotein to be able to do this, but all these up conformations of the RBD, which allow very efficient binding to a receptor like H2, and then puts the virus in this transmission window. So the SARS-CoV-2 is firmly in a transmission window, whereas SARS-CoV-1, which would be on the left, is less so. But there's a little danger to that in that this makes the spike protein relatively unstable, so the RBD can come off, the S1 can come off. So we're in this window of opportunity which allows this very transmissible virus. And as I mentioned already, this has been linked to. So is it firmly on the proponent of it's natural selection versus any kind of smoking gun and engineering? But this is what nature is able to have done. It's selected this spike protein with an insert, which has these properties. I'm sorry, my screen is moving slowly today for some reason. I don't know why. Why am I not moving forward? Okay, it's very slow. Apologies. All right, so and then what came out pretty rapidly at the start of the pandemic was when we started to grow this SARS-CoV-2 in the lab, especially in Vero cells, Vero E6 cells. The virus dropped that furin cleavage out very readily. So there were all these mainly deletions right in that region, but also some point mutations, which really, in my mind, adapted SARS-CoV-2 to grow very efficiently in furin cells, probably switched the entry pathway to a cathepsin prime pathway versus a cell surface temporis 2 prime pathway, but really stabilized the spike protein, and the virus in a way went back to its roots into a stable conformation in a protected cell culture environment where it didn't need to transmit from person to person, and went back to it's kind of happy place, if you want to call it like that, if viruses have feelings. Okay, slow screen. This is I have a new computer and it's never done this before. I'm not advancing. Maybe all I'll do, I'll escape and try again. I'm not escaping either. What shall I do? Okay, use the mouse. That's what I'll do. Okay. Right. So that being said, we're going to go into animal coronaviruses, especially this feline coronavirus FIP. So we're going to go. I'm going to take you down the rabbit hole of FIP and try and summarize this very intriguing feline disease in one slide. So there's a lot of text here. I appreciate that. So just bear with me. So it's a disease that was first described in the 1960s by an alum of Cornell, Gene Holtzworth, who was a very well-known feline medicine specialist down in Boston. And then, so it's a disease, a lethal disease of cats with a profile of typically effusion, so this so-called wet form of FIP, but also a dry form, which tends to be more neurological. Eventually, that was attributed to the coronavirus with these two profiles: a transmissible benign form, which is often referred to as FECV, feline enteric coronavirus, and then this highly lethal FIP form, the peritonitis form. And that, you can, I'm not going to go through all the text here, but there's a switch, apparently a so-called internal mutation, whereby in about 5 to 10% of cases, those FECVs will convert into the pathogenic form. And that's been a long-standing theory in the field. So it's not well understood, but the bottom line is that the FIP variants have macrophage tropism. So that's the key feature of like how the disease-forming virus can take hold. It gets into macrophages, triggers inflammatory responses and so forth, and that's the underlying mechanism. But it is also known to be really a persistent infection. A lot of cats will be shedding FECV for prolonged periods of time at different levels. It can be erratic. And there's also this phenomenon of ADE, whereby the macrophages can, the antibodies can drive infection. So a very complicated process, but not well understood, partly because the predominant clinical version, the so-called type 1 virus, is not generally isolatable in cell culture. So it's very limited. It's our knowledge of the basic virology of this thing. There's no vaccine, very difficult to diagnose molecularly. And then with the whole world of at least in the US, non-FDA approved treatments, including GS-like compounds and molnupiravir type compounds. And I'm not going to get too much into that today, but it's a big giant world of really the American Wild West is really happening in these cats. It's very much a Wild West situation. So the bottom line of all of this is that there's two biotypes, but there's also these two so-called serotypes. But what I hope to convince you of today is that's much more than a serotype. So if you look at a tree of feline coronaviruses, the red box up at the top, we have really two very distinct clades, or two very distinct genotypes. So the one at the very top is the predominant form, the so-called type 1 virus, and they're very, very different in amino acid identities. So the more I've always considered these to be very distinct viruses, the more we look into this, the more distinct they become. So even to the point of the disease process. And I'm starting to question, based on the original title or the original subtitle, whether the internal mutation hypothesis, as we know it, really fits with a type 2 virus. I think it definitely fits with a type 1 virus in a much more complicated way than we realize. And I'll get to some examples of that. But this mechanism of disease process may be fundamentally different with a type 2 virus. Okay, so the story starts, we're going back to actually the reason that influenza kicks in is that this was 2009 when we started to look at this, when it was the H1N1 pandemic, and we were interested in sequencing some samples, and we got contacted by our director of virology here at the diagnostic lab at Cornell, who had an outbreak, a so-called outbreak of FIP in a breeder, and we sequenced all these fecal samples, and we saw this furin cleavage site, what would appear to be a polybasic site, like would be in influenza HA. So this got me pretty worried at the time, because it was like, okay, maybe we've got a highly pathogenic virus circulating in this breeder. But it turned out no, that was one of the first surprises, that wasn't the case. So we went back and sequenced some more samples in the freezer, and this was typical. So this was present in all these non-pathogenic fecal samples. I should say these were not FIP cats. These were samples from the outbreak situation. So these were just surveillance samples from the breeder. So these were all non-disease cats shedding FECV with a furin cleavage site was what was going on. But that puts it very unusual for alpha coronavirus. If you look at this chart here of all the different coronaviruses, then it's the purple box at the top is it really stands out in the alpha coronavirus subfamily. Then no other alpha coronavirus has this. It's very quite common in the beta coronaviruses. SARS-CoV-2 would be one example of that. But if you look at the lineage A, the Embecco viruses, this cleavage site is actually pretty close sequence-wise to an Embecco virus, like the bovine coronavirus, an MHV-like sequence. So it's still a mystery to me where this virus came from, where this cleavage site came from. But if anybody has any thoughts on connections between an Embecco virus and this alpha coronavirus, I'd be happy to chat about that at any time, because it is intriguing me where this virus came from as a basic concept. And just at the bottom, just as a reference, is I'll be talking about some biochemistry. So if I start talking about P numbers for cleavage, that's the reference here. The P1 is the position where the protease will come in and make a cut, and that will then be activating the spike protein. So just keep in mind that the numbers go outwards from the middle, with a prime side and a non-prime side. Okay, so that was a starting point. And then what we decided to do about 10 years ago was to collect together all the fecal samples we have on the left, but then try and match that with some pathology samples. So we went to our pathology department and pulled some FFPE blocks, excuse me, and sequenced those. And then right away, we saw that there was a very distinct difference in that cleavage site. So as with those Vero cells with SARS-CoV-2, remember, not deletions, but point mutations were scattered through that cleavage site, and in what would be predicted to be the most important residues, P1, P4, P2 are the key residues. So that set us on a track, and I'll in the next slide, we've got many examples of this over a 10-year period now. I'll just show the last study just to summarize some of the possibilities. So we have the cat here, cat A, with a range of tissue. This is an FIP cat from an outbreak, a small outbreak from a local overcrowding situation, I suppose you might call it, in a home. So here we have a glycine mutation right in the P4 position. So that's standard through the cat. This cat is a little different. Different tissues will have different sequences. So it's the challenge here is like matching the sequence to matching the tissue, and there's never really any consistent pattern between different tissues and different sequences. It seems to be a fairly random process. And another example here of different tissue, multiple mutations, but different mutations in different tissues. So a big wide range of outcome really is the bottom line here. So really the challenge has been, how do we analyze that? How do we assess mechanistically what's going on? And that's where I'm driving toward, but also to note that it's not all about the S1H2 site. This is an early example of a cat where we sequenced S when it, one of those original samples from 2009. Then that cat subsequently got FIP 2 years later. So we've got a complete spike, and there's a lot of mutations in the spike. And the question here is, is this a reinfection? That's a possibility. Is it the virus has been persistent and is evolving over the years? So it's hard to tease this out. But there's the box in the middle there with the R to L mutation if you're in cleavage sites. So you can see here that we've got approximately 20 amino acids really involved in not only in the binding pocket itself in the purple and green boxes, but also in these yellow flanking regions. So the message I want to give here is that a furin cleavage site is actually a lot more complicated than it may appear. And what we're really aiming to do now is to go to a structural modeling lab down at Weill Cornell and really build these molecular dynamic simulations with structural information. This is an example we published back in the fall of MHV, and if you read the paper, you'll see that we can really map out exactly at a side chain level, like explaining why certain MHVs are cleaved and other ones are not. So this is a very powerful tool, a very sort of high-end computational tool to really to really inform what does this cleavage site entail and what's going on. This is back to that comparison of a SARS-CoV-2 with a very this is our own model, not a model from Harel, but you can see the P1 and the P4 are nicely separated and would get cleaved, whereas UU4 is a classic FIPV where the cleavage is not occurring, and structurally you can see even from this very simplistic model the accessibility of it's really scrunched up, and those key cleavage residues are really less available to the protease. So it's really a case of how is the cleavage site available to the protease, and trying to figure all of that out is still very much a work in progress. But that's what we're looking at. I won't spend a lot of time on this slide because it's just some examples of reference sequences in papers and in databases, and you can see at the bottom that we've got the red mutations or the classic mutations that would drop the furin cleavage down, but there's also a lot of these orange mutations which we're still trying to figure out. So that's still very much a work in progress, but this is more fine-tuning. So there's a lot of fine-tuning going on behind the scenes in terms of this transition, and that's, as I say, a work in progress. And this is where we really think that Harel's modeling can really help sort of sift through some of these sequences to look at how the fine-tuning's occurring and really how that all works. In my remaining time, I just want to mention one cat here, which I think is fascinating. This is now in bioRxiv. It's a cat that never had FIP but had this persistent infection. The fecal sample is a typical sequence, but then we've got some tissue samples which have downregulated cleavage sites. But the one I want to note mainly is this insertion here, which would be predicted to push this cleavage site out into space and make it dock more efficiently with the peptidase, with the protease, and enhance cleavage. And that's we're working through documenting that, but that's a pattern that does seem to be occurring. We get certain mutations, especially in conjunctival samples, where we got what are at least predicted at this point to be upregulated furin cleavage sites. So that's putting in my mind, a new transmission model for this virus. And maybe it's not really an enteric virus, it's more a respiratory virus. We just don't realize it. And maybe the conjunctiva are even super spreader events. And for those of you who know cats, they'll bump heads very, very readily. So it may just be a little sweet spot of transmission for cats. And this was actually back to our original paper. We actually didn't understand this at the time, but a lot of these flanking regions, those yellow residues or those orange residues are really potentially or actively upregulating cleavage, and maybe that's linked to transmission. So that's still a work in progress. And I'm just going to end with the news in terms of the big change in the FIP world since May has been the emergence of this FCoV23 in Cyprus. A lot of very important work. We're working very closely now with Christine in Edinburgh and the group in both Cyprus and various places to really figure out what's going on with this widespread transmission of an FIP virus in Cyprus, also linked to a so-called pantropic canine coronavirus in the Mediterranean region. And my real contribution to this is actually just came out just this morning in bioRxiv. So we've gone back into the freezer and really picked up a lot of these FCoV2 sequences, which we hadn't really been thinking about for a very long time. And there's one example here is it does seem to be that this CB05-like virus is not necessarily unique to the Mediterranean region, and CB05-like viruses were in the United States 10 years ago, in some cases causing like severe outbreaks, or at least localized outbreaks of, and this is a situation down here in in New York City in the Bronx Zoo, where some snow leopards got severe enteritis, and that the other sequencing of that's just been done, and that's now not a recombinant virus, but a pure canine, it seems, CB05-like virus causing this outbreak in these snow leopards in the Bronx Zoo. So I think there's a lot of work to be done in terms of tracking those viruses, and I'll make note of this, in that I'm quite personally very concerned about New York City, and when you look at the numbers of feral cats in New York City, it's staggering. Especially in Brooklyn and Staten Island, in these parks and cemeteries, and what's happening in New York City is that there's as many raccoons as people in New York City at the moment, and like the cats and the raccoons are hanging out together, and but it's a very concerning situation in terms of having seen what can happen in Cyprus. We want to really make sure that something equivalent isn't happening in New York City between dogs or raccoons or any other species, and really what's out there in the wildlife is still not clear. So that's, again, more work in progress, and it's going to keep us busy for some time. And I'll just end with this one slide here of like trying to link together the three viruses I've talked about: SARS-CoV-2, FCoV1, and FCoV2. And the new aspect here is the work started out with Tom Gallagher on these NTD antibodies that regulated crosstalk, and that's one of the key things with FCoV23. So it's missing its zero domain. So we're now going back to FCoV1 and actually looking more closely at the zero domain there, and clearly there's some changes. That's the NTD of the 234 sequence from many years ago, but also there are examples of FCoV1s that are missing zero domain. So it's set up a whole new process of investigation to connect together the zero domain with the cleavage side and the whole fusion activation process. So that's the update, and I'll end there and thank a whole bunch of people. I won't go through everybody now, but I do want to thank Jimena, my postdoc, who's really driving a lot of this sequencing activity that we really depend on for trying to understand what's going on here with all these different viruses. And I'll stop there.